All right, so thank you very much to the organizers for the opportunity to tell you about the work that's going on in my lab. So we are a lab that develops technologies for high-throughput functional genomics using the CRISPR system. And we've been applying them in a number of areas, but with a particular focus on trying to identify drug targets in cancer and understand how cells take things up from their environment by endocytosis and phagocytosis. So what I'm going to try to do today is to tell you three short stories. The first is about a technology that we've developed to look at pairs of genetic interactions, in particular to look for interactions between drug targets and cancer cells. I'll describe a new technology we've developed to create localized populations of diverse mutants in the genome with the purpose of enabling directed evolution experiments. And finally, I'll tell you an unpublished story in which we've developed a strategy to look in high throughput for genes that control the process of phagocytosis. We make use of a platform which is by now pretty widely used, which is to create custom libraries of oligonucleotide synthesized guide RNAs, which we transfer into cells using a lentiviral library. We then screen these libraries in pooled formats and then read out the hits using deep sequencing. And so most people are probably familiar with this by now. We use a very large population of negative controls against which we compare gene targeting guides to identify hits. And then at the end of these screens, we and everyone and their mother who's doing these screens now end up with a laundry list of genes. And so to try to understand how these genes actually interact, we borrowed a technique that was developed in yeast by Charlie Boone, Brenda Andrews, and Evan Krogan and others to look at pairwise genetic interactions in very high throughput. And so to do this, we create genetic interaction maps where we knock down pairs of genes. And so in these maps, you have a phenotype of a single gene in combination with a set of second genes, and the positive and negative interactions give you a pattern characteristic of the function of that gene. You can then classify genes based upon the similarity of that pattern to get genetically related genes and classify newly uncharacterized genes. And so one of the things we're most excited about is to use this technology not just to classify genes that were uncharacterized, but to look in very high throughput for synthetic lethal combinations in cancer. And so this is, of course, a dramatically important problem in the context of cancer drug development, where you have this problem of drug resistance, which characterizes all these fantastic new drugs that are being developed. So there's this now famous example of a melanoma patient who presented with this fairly dramatic disease, which a fantastic drug against BRAF was remarkably effective. But then, as is inevitably the case, you have resistant cancers that develop. So after a period of time, the same tumors come back. And so the problem is cancer drug resistance. And so if we could find a second drug that would work in combination with this first drug to prevent this escape, you could potentially dramatically improve therapies. And so, of course, to do this, creating new drugs is very expensive. And so we wanted to actually, if we could, repurpose FDA-approved drugs, which would be great. But then even if you had those and you wanted to test all pairwise combinations, this would be very slow. And so the idea we had, which was to, instead of using combinations of drugs, we would target the targets of those drugs using CRISPR and pairs of guide RNAs. And this was the work of Kyo Han, who's a fantastic postdoc in the lab, together with Edwin Zhang, a graduate student. They got help from David and Amy. And the idea was to do this in very high throughput. So can we take 500,000 guide RNA pairs, targeting combinations of drug targets, and look for these rare synthetic lethal combinations? And so I'm going to skip over an enormous amount of work that was done by these guys. This was published last year. And suffice it to say, we developed a strategy we're quite comfortable with to express pairs of guide RNAs. It turned out to be much more complicated than anyone could have imagined, just to express them in a way that didn't get tons of recombination and could be paired with deep sequencing. But we and many others now, including Trey Eidecker, Prashant Mali, and Andrea Ventura, and others, have come up with a strategy for expressing pairs of guides and doing these genetic interaction maps. So what we did was we took guide RNAs targeting drug targets. We selected genes which had a mild phenotype, but which were not totally lethal by themselves to maximize the opportunity to find synergy. And then we created a large pairwise interaction map composed of these targets. And then the workflow is exactly the same as for the single gene screen, except now we have ligated pairs of guides, which we then infect into cells and select for genes that selectively drop out only when knocked out in combination. And so just to kind of show you some quality control for this data, replicates are very highly reproducible, which is comforting. And it doesn't matter which position you have the guide in. If you flip A and B, you get the same phenotype. So these are highly reproducible as well. And what you get from this, it looks like this. So what I'm showing you here is the genetic interaction score of one replicate on this axis. This is the difference between the measured phenotype of the double mutant and the expected phenotype from the two single mutants. And the second replicate on this axis. And what you can see is that by and large, the vast majority of these gene pairs have no genetic interaction at all. And this, in fact, is not that surprising if you think about it. Most genes don't actually have too many synthetic lethal interactions. What was comforting is if you knock out a gene and then you add a second guide RNA against that same gene, this phenotype of the double mutant is buffering because once you've knocked out that gene, there's no further cost to knocking it out again. So as you'd expect, it's better than you would have predicted. But what was satisfying is we could actually find rare but quite potent combinations of genes which, when knocked out together, exhibited potent synthetic lethality, including several druggable targets. And we decided to focus on these two genes, BCLX long and MCL1, which were two anti-apoptotic members of the BCL2 family. And you might say to yourself, well, of course, these genes are synthetic lethal. But in fact, members of the BCL2 family vary enormously across different cancers in the degree to which they are actually lethal. So what we did was we took the corresponding drugs against MCL1 and BCLX long, and we could show that in combination they were much more potently toxic than either of them were in combination, singly, I should say. And this was great. We could see this in K562, which is a BCR-able CML line. But it was nice to see that they were not just globally toxic. So we added these two drugs in LCLs or in primary CD34 positive hematopoietic stem cells, and we really don't see very much synthetic lethality at all, which was comforting because you don't want to just kill everything. But what was even nicer is if you create an imatinib resistant K562 line, you can see that these two drugs are even more synthetic lethal in that background. So in some ways, K562 is one of the least interesting cancers to solve because there's already a fantastic drug, imatinib, but still in about 10% of cases you get imatinib-resistant cancers that come up. And this combination seems to be actually quite effective in that background. So we're actually quite excited about this technique. And in general, we were satisfied to see that you could start from a gigantic library of guide pairs and pull out what seems to be a validatable drug-target combination that works in this cancer. And we're now interested in applying this broadly in a number of different cancers. Okay, so that's basically a summary of what I just showed you. And in the next little bit, I want to describe a new technique that we developed to create localized point mutants in the genome. So typically, if you think of mutagenesis with Cas9, you're thinking about the use of active Cas9 to basically, as George Church would say, vandalize the genome by creating large holes in it or create indels, which create stop codons and delete gene function. But in some cases, you might actually like to install specific point mutations to create particular mutants. And so a number of groups, including the lab of Jason Dury, have shown that you can take a guide RNA and a library of single oligonucleotide donors containing a single nucleotide mutant and install those in that position in the genome. And so this is nice. You can actually do this. But of course, this requires that for each mutant you want to put in, or each library of mutants, you have to have a new guide RNA and a new library of oligo donors. So what we wanted to do was to come up with a way of creating diverse but functional mutations in the genome in situ. And so to do this, we turned to the way it's done in nature. So with remarkable precision, during the process of antibody somatic hypermutation, your body has a way of creating mutations only at the diverse region of the antibody. And so nowhere else in the genome, but only exactly in this diverse site in the genome. And so this is done using the enzyme AID. And so a very talented postdoc in the lab, Galen Hess, decided he would use dCas9 to recruit AID to positions in the genome. And with a number of sort of engineering steps to get a hyperactive variant of AID, he could show that with a single guide RNA, you could create a pile of point mutants exactly where you put the guide. And so what does this look like? So you actually you heard a very nice talk from Nozumu, who showed that you could have dCas9 recruiting a different cytidine deaminase. And this, from the Kondo lab and David Liu's lab, you could create a C to T conversion. So this is fantastic if you want to create that precise change. And in diseases where you should have T and it's a C, you could imagine this therapeutic editing. But what you're seeing here is that this hyperactive variant of AID, with some bias towards C and G, of course, is creating substitutions to every other possible base. So you're getting, instead of C to T conversion, you're getting base diversification at this locus. And so within a range of about 100 base pairs centered on the PAM, you can get diverse point mutants in the genome. So now you can do neat things with this population of mutants. So for example, we could take guide RNAs targeting wild-type GFP, and with a single round of mutation and flow sorting, recover this S65T mutation, which is the known mutation that creates eGFP. More interestingly, we could actually target the entire protein coding region of PSMB5. This is the chemotherapeutic, the target of the chemotherapeutic drug bortezomib. We can mutagenize the coding region and then select with bortezomib. And only in a library that targets this protein, but not a control library, do we get enrichment of these mutations that confer resistance to bortezomib. So this was neat, because now you can take the structure of PSMB5, for which the binding site to bortezomib is known, and you can see that we were able to recover mutations that map exactly to the position of this binding site for the drug. Some of these had actually previously been demonstrated in patients that have been treated with bortezomib, but some of them are new, and in particular, we could identify new mutations on the complete opposite side of the protein that nonetheless conferred potent resistance to bortezomib. So we're very actually excited about using this technology to map drug-protein interactions and understand that not only the binding sites, but potentially modes of resistance and allosteric sites on drug targets for which you might be able to improve for the design of different drugs. So we're very excited about this technique. We've been continuing to improve it in a number of different ways, which I'm happy to discuss with people. We're also happy to share these tools in case anyone would like them. And we are now applying them in a number of different areas, including drug target identification with different academic and pharmaceutical collaborators, but also for the sort of native purpose of using AID, which is to create diverse libraries of antibodies, which with an in-cellular assay, we can actually couple to functional readouts. And this simple system, of course, requires only a guide RNA, but not the homology donor. And of course, it goes without saying you can create diverse mutations at multiple locations in the genome and potentially look at things like coevolution at 2 different sites. Okay, so in the last bit, I'm going to tell you about a new project in which we've been developing a strategy to understand how cells take things up by phagocytosis. So traditionally, phagocytosis is how the immune system would recognize things like bacteria, engulf these large particles, and clear them from the body. But over the number of years, and particularly recently, phagocytosis has been seen to be important in a number of diverse processes, including synapse pruning during neuronal development, tumor surveillance, turnover, of course, of senescent cells, and in neurodegeneration, failure to phagocytose protein aggregates and debris is potentially thought to be important in neurodegeneration. And so the question that we set out to ask was, how do these macrophages, which exist in every tissue in your body, recognize and engulf very diverse particles like these and maintain cellular homeostasis? So to do this, we developed a strategy based on magnetic separation, where we took a monocyte cell line, which could be differentiated into macrophages. We introduced our CRISPR library. We then fed these macrophage cells magnetic beads and passed them over a column, where the idea is if the cell has eaten a magnetic particle, it will stick to the column. If you knock out a gene required for phagocytosis, it will flow through. And so you can very quickly separate cells which can eat from those which can't, and actually, of course, find things which can eat even better. And I should mention, this is a screen which can be done in about half an hour, as opposed to normal growth screens, which of course take several weeks sometimes. And this is the work of Michael Haney, who's just a fantastic grad student in the lab, and it was a wonderful collaboration with Chris Boleyn, who was a postdoc in Ben Barris's lab. And they got help from David, Kim, and James, and a number of others. But the neat thing about this technique was, so not only can you feed cells beads of different types, different sizes, different charges, different shapes, actually, but you can then magnetize many different other particles. So you can take these magnetic nanoparticles, cross-link them onto red blood cells, myelin, thymosin, protein aggregates, and things, and then compare the requirements for uptake of these different particles using the exact same system. And so just to kind of give you a flavor of the types of things we recover, as expected, you get all of the actin machinery that's required for this really remarkable plasma membrane deformation process that allows the engulfment of these large particles. So we quantitatively recover the entire scar wave complex. And then, interestingly, we recover many other genes that really had not been previously shown to be involved in phagocytosis, including lipid biosynthesis genes, some involved in vesicle trafficking, and a number of genes with unknown function, a few of which I'll describe to you now. But with this data set, so we now have actually even more than this, but 8 different genome-wide screens and the requirements of each of these particles. And so what you can see is that while some particles, for example, the actin machinery is required for uptake of all particles, but then there are certain particles for which you see a selective requirement of certain genes, but not others. And so we're very interested now in mining this enormous data set to try to understand the function of genes that seem to be selectively required for uptake of certain particles. So just to kind of show you a couple of examples of this type, we could find that this gene, TM2D3, which was very poorly characterized but had been previously shown by GWAS to be associated with late onset Alzheimer's disease. When you knock out this gene or several other components of what is now known to be a physical complex of TM2D1, 2, and 3, you can see selectively they seem to, so I should say, wild-type cells take up these 2 different particles, synaptosomes or A-beta aggregates, which are important in neurodegeneration, about the same. But when you knock out this gene, you can see selectively they're unable to take up A-beta aggregates, but they can take up synaptosomes just fine. So we think this is potentially an explanation for the pathogenesis of this gene in Alzheimer's disease. If you fail to take up A-beta aggregates, this would be sort of worse for the disease. And in the last little bit, I'll tell you about our new favorite gene, NHLRC2. So this gene exhibited one of the strongest phenotypes in any of the screens that we did. The only things that were nearly as strong were the scar wave components of the actin reorganization complex. So this gene was very poorly characterized, but when you knock it out, it basically blocks uptake of every single particle that we tested. To identify interactors of this, we did bioID, so we tagged it with Beret, and we could see that physically it seems to associate with some known actin regulators, including this P190 ROGAP protein. So we had a suggestion that this gene was involved in actin organization based on the strength of its phenotype and these physical interactors here. But what was even more convincing is this experiment. So what I'm showing you here is the time course of uptake of Frodo-labeled particles. So these fluoresce once they reach the low pH of the lysosome. And what you can see here is when you knock out NHLRC2, as I mentioned, you completely block uptake. But if you overexpress in this background a constitutively active RAC1 protein that sort of activates the actin machinery downstream, you can basically completely bypass the requirement for NHLRC2 and restore phagocytosis. So this seemed to be quite convincing, a case that it was indeed a new actin regulator. And so the model is something like this. NHLRC2 is an interactor of P190 ROGAP that seems to regulate this upstream actin regulatory machinery. When you knock it out, you have a defect in actin reorganization. But then we did what was one of the coolest experiments that I've done in the lab, or it wasn't me actually, this was Michael Haney, but it was one of the cooler days in lab in quite some time, which we actually went down to the scanning electron microscopy facility and looked at what these cells look like under the microscope. And so this is a wild type scanning electron image of raw 264 macrophages engulfing a bead, and you can see these spectacular lamellipodia that sort of reach out and engulf these gigantic beads. And then all over the surface of the cell, you can see they have these spiky projections. These are filopodia that the cells use to sort of scan their environment and which are also involved in the process of phagocytosis. And the rest of these images are just sort of gratuitous pictures of phagocytosis. So these are, again, wild-type cells, actually the same image as before, but you can see all the wild-type cells have these, they're covered in these filopodia. And then, here's another wild-type cell trying to engulf this gigantic bead. But if you knock out NHLRC2, you can see that while you can occasionally find filopodia, for the most part, these cells are completely devoid of those surface actin structures, these projections. And in fact, they also have this interesting lack of cortical actin organization, which results in the plasma membrane kind of spilling across the surface of the plate here. So yeah, this is quite interesting. And so these are just more images of that same cell. They do have these sort of surface, strange-looking whorl structures that actually do not contain actin, we discovered. And these cells will occasionally contact a bead, but they will not complete the process of engulfment. So yeah, we still have a lot to understand about how this gene works, but we think we have a new actin regulator. So this is just staining with phalloidin for the same actin structures. So to summarize this, we've developed a strategy that we think is actually quite broadly applicable for studying the process of cellular engulfment. We've developed through this effectively an almanac of all the genes that are required for phagocytosis of different particles. We found genes that are general and also selective for particular particles, including several new genes that seem to be important potentially in Alzheimer's disease and as a regulator of the actin machinery. So with that, I'll end. There's a number of people to thank for this work. So Kyo and Edwin built the double guide platform that I described to you. Galen Hess is a postdoc who developed this CRISPR-X machinery for creating mutations. And then Michael Haney, together with Chris Boleyn, did the phagocytosis project. And with that, I'll take any questions. Thank you.
Wonderful talk. Do we have any questions?
Great talk. I was wondering, for the phagocytosis screen, did you find genes that were upregulating phagocytosis, and what organelles or associations are those?
Yeah, so this is a really interesting question. So yes, we found many genes that when you knock them out seem to increase phagocytosis. And you can imagine this would be interesting in the context of neurodegeneration or something where you want to actually increase clearing. We found a number of different categories of genes, and particularly genes that seem to be required for synthesis of sialic acids on the cell surface. Typically, those are known to be important on target cells for blocking recognition by the immune system. And so we're interested in trying to understand why is it that on the macrophage side, the regulation of sialic acid synthesis could be important in particle recognition. There are a number of other genes for which we're not really sure about their function. Some of them are actually like negative regulators of the actin machinery, so those make sense in the context of what I showed you. But there's a lot of interesting stuff there.
So going back to the first part, where you were looking for synthetic lethality, but you also showed that you could get, quote, buffering. Have you looked for drug-drug interactions that actually lead to better growth? Because that also informs people what not to use as drug combinations.
Yeah, this is a really great question. So, and what we're trying, I should say, we're trying to develop models where we can better recapitulate increases in growth rate. One of the most striking things about doing any of these screens in highly transformed cell lines is that the vast majority of genes have zero positive effects at all. Most of them have a negative effect on growth. So it's actually really hard to find anything that makes K562 cells or many other cancer cell lines grow faster, because they're already sort of maxed out. There are about 10 to 20 genes in the genome that do that. These are negative regulators of mTOR complex, mostly. So we did see some of those in the context. We could see buffering interactions in the context of mTOR negative regulators, but it's hard to find those under the standard growth conditions. We're trying to now model a little bit more accurately, using 3D culture, for example, tumor growth conditions where you see more of those positive growth phenotypes. But in this system, there's just not that many of them. Yeah.
Thank you, Mike. And let's thank all speakers in this first session. Please talk to us during the break if you have more questions, and be back in 20 minutes, I think.